because usually I just forget. All right, it's 3 p.m., so we only have 6% for the moment, but it's time. So let's start. I'm sure in 1 or 2 minutes, we'll have more people. So good afternoon, everyone, and welcome back to our research and winemaking webinar series that started last year, but we decided to provide more information to you again this year with more specific topics. And so we already had the enzyme selection and yeast selection the past 2 months. And so today that's going to be about microbial spoilage. So for those of you who don't know me, I'm Dr. Aude Watrelot. I'm an assistant professor of enology in the Department of Food Science and Human Nutrition at Iowa State University. And I'm co-hosting this webinar with Drew Horton, who is a research specialist of enology at the University of Minnesota's Grape Breeding and Enology Project. So just before we start the webinar, here are some instructions. I'm sure you already all know about those instructions, but just to make sure, so the webinar is being recorded, and if you are interested in watching again the previous recordings of last year or some of the session of this year, that's available on YouTube, and I'm going to put the link into the chat box throughout the presentations. Then when you have a question, feel free to use the chat box or the Q&A box. If you don't see one of them, you should have 3 dots at the bottom of your screen. So click on the 3 dots, and then you should have access to the Q&A box. And also, I'm recording this webinar. And at the end, you're going to have a link for a survey. So please just take 5 minutes to complete the survey, because we are always looking for feedback to improve the tools we provide you. So today it's our great pleasure to have Maureen Moroney from the Midwest Grape and Wine Industry Institute and Dr. Chris Curtin from Oregon State University, who will present theory and practical aspects about microbial spoilage, how to manage the microbial spoilage risk, or how to manage everything in the winery and in the wine. So that's to provide you all the information you may be looking for, especially for harvest. That's going to happen very soon if it didn't started yet. So thank you very much, Maureen and Chris, to be with us and for accepting our invitation. And I'm leaving Drew introducing Maureen. Thank you.
Thank you, Aude. It's my great pleasure to introduce Maureen Moroney. Maureen is a research scientist at the Midwest Grape and Wine Industry Institute. She studied biology at Iowa State University, and she spent 8 years in production winemaking in Sonoma County, California, before joining the Institute in 2016. Her current roles include managing the fee-for-service wine lab at Iowa State, participating in research and research winemaking, and providing excellent educational programming to the industry, programming just like you're seeing today. She is also a graduate student in interdepartmental microbiology, focusing on naturally occurring vineyard yeast. Today, Maureen is going to present for about 25 or 30 minutes, and if you have any questions, we will get to them at the end of the program.
Thank you, Drew, and thank you, Aude. And thank you, everybody who's watching this. Hopefully you'll get something out of it. Hello, everybody. I guess this is the part where I try to share my screen, right?
You should be able to.
Okay. All right. Share. And how is it looking for everyone?
It's
okay.
perfect.
Sweet. All right, yes. So I am, as Drew said, I'm a research scientist at the Midwest Grape and Wine Industry Institute. My contact info is listed here at the bottom if anyone has questions that they want to follow up on later. So I'm really pretty briefly going to do sort of the 30,000-foot view of microbial spoilage in wine. So starting at the very beginning and then leading into something more research focused in the second portion of this webinar. Oops. See if I can actually advance the slide. There we go. All right, so starting very basic, what in the world is microbiology anyway? Micro meaning small, bio meaning life, and logi or logi having to do with knowledge. So microbiology would be the study of very small living things. But when it comes to wine, we are mostly concerned with two classes of microbes, so bacteria and fungi. Fungi, of course, includes yeast. And yeast, as we all know, is really important for wine. But when we think about doing microbiology, most people probably picture using a microscope or looking at colonies of microbes on little agar plates. And those techniques are still widely used and they're still valuable. But there are also lots of other ways to gain insight into what the microbes in our wines are doing. And some of them are things that a lot of us do on a regular basis without really thinking about the microbes behind them. So, you know, in addition to doing, stuff with a microscope, doing stuff on plates, maybe even doing genetic testing where, we're finding out what types of genes are present in the wine so we can relate that back to the microbes. But beyond all of that, we can measure the chemicals that are being consumed or produced by the microbes. So that means focusing on the metabolic processes of those microbes. And to put that in terms that a lot of us are very familiar with, in winemaking, some very common chemicals that we measure are things like sugar, alcohol, malic acid, and acetic acid. And all of those things can be either consumed or produced by wine microbes. So they tell us something about the microbial activity that's going on in the wine. So why are microbes important for winemaking? Probably the biggest reason is that we can't have wine at all without them. So some of the major components, things like alcohol and lactic acid, are things that are produced by microbes. But additionally, we can see that there's all types of other metabolites that are important, things that are related to aromatic compounds and other secondary metabolites. So yes, why are they important? Well, because they make the wine, right? They make the wine what it is. And we know that wine yeast converts sugar to alcohol, right? But as you can see here in this diagram, it does a whole bunch of other stuff too. And many of those pathways intersect with each other. Some of those pathways produce things we like, and some of those pathways produce things we like, and some of them produce things that we don't like. So in order to understand spoilage, we have to kind of understand what we consider a good wine microbe. And really a good wine yeast is one that has been selected over centuries based on its positive attributes. So in wine, we typically use a species of yeast called Saccharomyces cerevisiae. And that's the same yeast that's used to make beer and to make bread. But even within that single species, there's variation between individual strains. You may be familiar with choosing commercial strains of Saccharomyces cerevisiae wine yeast based on what they're supposed to do for your wine. So I pulled this table. Probably some of you recognize where it came from. A certain supplier of winemaking additives, supplies this table that shows, okay, what are the characteristics of the yeast? What's its alcohol tolerance? What are its nitrogen needs? What's its fermentation speed? Things like that. And so these different strains were developed by selection over time because of those positive attributes. Or here on the left, we have, 3 hypothetical yeast strains that produce different aroma profiles in the wine. So they pull out different attributes from the chemistry that was already there, and they release different aroma compounds. So yeah, things like boosting certain aroma profiles, alcohol tolerance, nutrient needs, even compatibility with malolactic bacteria. These are all traits that can differ from one yeast strain to the next, even within that species. And then looking at ML bacteria, just like with yeast, we know that ML bacteria convert malic acid to lactic acid, but similarly, they also do a whole bunch of other things along the way. And again, just like with yeast, we have our typical good wine bacteria, which is a species called Enococcus eni. And there are different strains of bacteria within that species too. They all do malolactic conversion, but when it comes to all those other pathways, some strains may favor some products over others. Enococcus eni, which is our malolactic bacteria, is one species within a broader category of lactic acid bacteria. So other lactic acid bacteria, or sometimes you'll hear me say LAB, other LAB include species of Lactobacillus and Pediococcus. And somehow diacetyl always comes up when we talk about ML bacteria, so I want to take a very brief moment to clear up some confusion about diacetyl. You can see it here on this metabolic pathway. You can also see that the starting points are sugar and citric acid. So the notion that diacetyl is primarily a product of conversion of malic acid to lactic acid is not 100% true. You can also see that diacetyl, which is that strong buttery aroma, is a possible exit along the way to, at the end of that pathway, there's a gentler creamy aroma, which is the actual lactic aroma. So while there are some ML strains that may favor diacetyl production more than other strains, it can also have to do with timing, where the sugar and malic acid have been consumed before citric acid metabolism begins. Maybe the wine gets sulfited and the pathway never makes it to that final endpoint. So the gist is it's super complicated. There are a lot of possible causes and a lot of possible outcomes of our various aroma compounds in wine, but blaming your ML bacteria or your ML conversion for your diacetyl production, it's not quite as simple as that often. But getting back to the topic at hand, what is microbial spoilage? We've looked at now, very briefly, at the normal function of quote unquote good wine yeast and bacteria, but we also know that that normal function has quite a bit of variability within it. So that brings us to microbial spoilage. What is it and what causes it? You will probably not be shocked to hear that it is complicated. Most spoilage microbes produce multiple aroma and flavor compounds, and many fault compounds can be produced by multiple different types of wine microbes. So here, for example, you'll also see that our friend Enococcus eni is included here as a type of spoilage bacteria, for example, with the potential to lead to things like geranium taint. Now, when we talk about sensory faults in wine, we define them in contrast to desirable attributes. Faults are really simply just unwanted sensory characteristics. And faults can come from multiple sources, not all of which are from microbes. But there are many sensory faults that can be caused by wine microbes, including things like cork taint, Brettanomyces, you're going to learn a lot about that, or the Brett character, the Brett or sensory character, volatile acidity, which is that vinegar aroma, ethyl acetate, which is the nail polish aroma, acid aldehyde, which is associated with an oxidized characteristic, so like bruised apple or sherry or nutty aromas, lactic spoilage, so that kind of sauerkrauty flavor, also unwanted or unhealthy yeast fermentation or ML bacteria, you know, when good microbes go bad, right? Or even visual effects, so things like browning and haze. And so you'll notice here that I underlined unwanted or unhealthy yeast fermentation or ML. This is really important because, again, even our good microbes can do things that we don't want them to under certain circumstances. So from, you know, a practical perspective, how do we prevent microbial spoilage from leading to a faulted wine? And sort of working through this in roughly chronological order of production, good Good quality fruit is super important. If you're bringing in damaged or diseased fruit, that means you're already starting at a disadvantage against things like acetic acid bacteria. We all know that winery sanitation is critical. If you can keep your wanted microbes healthy, that helps them do what we want them to do. And it can also help keep populations of unwanted microbes in check. There are some chemical parameters that we can control in wine. We can also control some environmental conditions in the vessel or in the winery facility space. In order to make sure that everything is going well, we need to be routinely testing and tasting the wine. And if it turns out that things aren't going well, we have some options for getting rid of unwanted microbes. But as a very last resort, we might be able to fix some sensory problems after they happen, but that's definitely not an ideal outcome. In the next few slides, let's actually take a look at some of these practices and what that means. We know that sanitation is so incredibly important in the winery. It's also a huge topic all by itself. I really can't do it justice in the time that we have today, but if you're interested in more detail, there is a previous video in this webinar series, and I highly recommend checking it out. There's a recording on YouTube for the winery cleaning and sanitizing webinar. It's available on the YouTube channel. It's from last August, so please check that out. But the very abbreviated version is that there's no one size fits all prescription for cleaning and sanitation, because every winery has different needs, different equipment that they have available, and other factors like that. With that said, whatever products you are using should be used according to the supplier's instructions. So this includes things like the concentration, the temperature, the contact time, and those other details. Physical removal of grime is the necessary first step before trying to sanitize anything. Sorry, I sort of lost my place. Oh, yes. Okay, so before trying to sanitize anything, yes. And just like with antibiotic resistance in human medicine, microbial populations in the winery can build up resistance to sanitizers. So rotating sanitizing products is good practice to help avoid that. Some parts of the winery are simply harder to clean than others. So this includes things like valves, rubber fittings, anywhere else that's hard to reach with scrubbing action or where liquid can pool up. Particularly for wine in barrels, it's really important to be aware of the risk of cross-contamination from one vessel to another. And as we mentioned earlier, quality wine starts with quality grapes. So we want all our equipment to be as clean and well sanitized as possible. Does that actually mean that it's completely sterile? I would say the answer is definitely not. In the winery, it's really impossible to completely eliminate microbes. So personally, I like to think of wine as a medical patient that I'm trying to keep healthy. We're trying to prevent the patient from getting sick. In medicine, they use what's called the disease triangle, which I have shown here. And in order for a disease to occur, you need three things. You need a susceptible host, a favorable environment, and the pathogen. And in this case, this is going to be a spoilage microbe for the wine. And if you have any two of those, but you don't have the third, the patient won't get sick. So that means we can come at spoilage prevention from multiple angles. But you know, let's actually consider a real world example using that disease triangle as applied to winemaking. One of our most common wine spoilage microbes is acetic acid bacteria, which produces that vinegar aroma we talked about. And I know that you can't answer me out loud, but you can answer each of these questions in your head. So are acetic acid bacteria present in your winery? Is wine susceptible to acetic acid bacteria? Is wine a favorable environment for acetic acid bacteria? And unfortunately, the answer to all of these questions is yes. So we actually can't eliminate any of the 3 points of the triangle. So now what? Well, maybe you've seen this, or at least a very similar image to this. I made some modifications. But the idea here is that in winemaking, to prevent spoilage, we have to use as many strategies as we have available because none of them are perfect. When it comes to preventing wine spoilage, it's all about reducing risk. So the more strategies we use, or the more layers of defense that we have, the less likely that our winery microbes can get out of hand and cause a problem. In the winery, this means 3 basic things. So we want to reduce the number of bad microbes through things like sanitation, filtration, and early detection. We want to keep the environment unfavorable for spoilage through things like environmental control and promoting healthy microbial growth. And we want to keep the chemistry unfavorable for spoilage as much as we can. So that means controlling things like pH, SO2, alcohol, and sorbate, or at least measuring them and being aware of them. Another note, of course, is that dry wines tend to be lower risk. If there's no sugar to metabolize, there's a much lower risk of spoilage. But because none of these can be achieved perfectly, we need to use multiple strategies to minimize our risk. I'm highlighting that repeatedly because if there's one thing I want you to take away with you today, I want it to be that. So coming back to something I mentioned earlier, we want to take care of our good microbes. That's actually a really important part because, if we don't have a healthy microbial population, there's basically wide open space for spoilage microbes to take over without any competition. So we want our good microbes to be healthy and do their jobs, but we also want to use them as a strong offense as part of our good defense against spoilage. So this means making sure that you give them proper nutrients. You want to avoid extreme temperatures that can kill them or make them sluggish, and all those other normal things that you want to do to facilitate healthy growth. In addition to promoting good microbial growth, winemakers also have chemical tools in their toolkit to make a wine a little less hospitable to spoilage microbes. So I consider this kind of like the susceptible host point of the disease triangle. In terms of chemical strategies, sulfur dioxide is absolutely a critical one. And once again, that's a whole topic by itself. And there is a recording of that webinar too. But things like a low pH and a high alcohol level can help inhibit spoilage microbes. And once again, dry wines tend to be at a lower risk than ones with residual sugar. So that third item, the environmental strategies, we can make the environment less favorable for spoilage microbes. So our main tools here, I would say, are low temperatures and minimal oxygen exposure. So whether that means keeping vessels topped or using inert gas to exclude oxygen, those are the main things, is temperature and oxygen exposure that you want to keep control of. Okay, so we're doing what we can to prevent microbial issues, but how do we know that it's working? We have to be really proactive about monitoring the wine, and that means both testing and tasting. So we talked about chemical testing for things like sugar, alcohol, malic acid, and acetic acid. Those are good ways to keep tabs on what the microbes are doing. And we also need to add in the parameters from our chemical prevention strategies, which include SO2 and pH, as well as sugar and alcohol again. Biological testing is really less routine for, I would say, most winery labs until you get up to the really large sizes. But if you suspect a spoilage issue, I would recommend that for sure on a case-by-case basis. It'll help you troubleshoot it. Or if you really want to audit your process by doing things like, checking sterility in bottled wines post-bottling, then you can be looking at biological testing strategies, I would say. And for sure, every good winemaker should be tasting their wines on a regular basis for lots of reasons, not just to monitor for spoilage, but because you should be tasting your wines on a regular basis if you're a winemaker. So if you're doing those monitoring steps and you discover that there is an issue, Or if you anticipate an issue in the future, maybe with a wine that's currently dry, but maybe you're going to sweeten it before bottling. There are some issues, or some options rather, available for dealing with unwanted microbes, and they fall into three basic categories. Those categories being inhibition, killing action, or removal. So to talk about some of the more commonly used strategies, sorbic acid is commonly used as an inhibitory compound against yeast re-fermentation in sweet wines, but it can actually be metabolized by lactic acid bacteria and produce geranium taint. So not a silver bullet. Similarly, lysozyme, not a silver bullet. It can be used to kill unwanted lactic acid bacteria, including ML bacteria, but it does not work on yeast. There is a compound that you may know by the name of Velcorin, which is actually pretty great for keeping wines microbially stable in the bottle. The downside here is that it's often not an option because it requires specialized equipment and handling. And sterile filtration is a great and commonly used practice. It can be used to physically remove microbes from the wine, but only when it's done properly. And this assumes that all of the downstream equipment is also thoroughly sanitized. If you've had an issue with microbial spoilage, and let's say you've gone ahead and killed or removed the microbe responsible for that spoilage, the thing to remember is that the chemical compound will still be present in the wine, and vice versa. If you remove the chemical compound but you don't remove the microbe, they can just make more of it. So let's say, okay, you killed or removed the microbe responsible, the chemical compound is still present in the wine, but if you caught the issue early, maybe the levels of that chemical are still low enough that it's not really a problem. If so, great. On the other hand, if the levels are high, you may need to remediate it somehow. So options there include removal technologies, some binding agents, depending on what the issue is. It's always an option to blend into a larger batch of wine to dilute the concentration to an acceptable level. Unfortunately, once you reach this point where you have a problem, none of these options are really very good ones. So absolutely, if possible, your goal should be avoiding getting to this point because once you're here, none of the outcomes are really ideal. So that was running through a whole bunch of hopefully a refresher. Hopefully you're already on top of most of that, but it's good to revisit it. So I've talked a lot about the options that we have. I've actually used that word options quite a lot. And that's because I know that every facility and every batch of wine is different, which means there are no universal rules to give you. The one thing I can tell you is that there are some basic best practices, and they involve controlling to the best of your ability those three things we talked about: sanitation, environment, and chemistry. And because your specific procedures will be unique to you and your facility, that means you are the one responsible for making sure that they work. The only way to find what works for you is to commit to monitoring your wines throughout the process, both in the lab and in your mouth. So with all that in mind, I think I just want to leave you with some questions. And these are questions that I think our next presenter will hopefully help you answer as you think about them. But let's say hypothetically, hopefully none of you, this is the case, but let's say hypothetically you have a wine that you think smells like it is faulted with Brett or Brettanomyces. So I want you to think about, or I want you to consider things like, okay, what is Brett? What does it do? What information do you need to gather before your next steps? And what actions should you take? Also, how can you avoid issues in the future? So I thank you for your attention, and hopefully you're all having a good afternoon. And I'm really, really looking forward to hearing our next presenter. But thank you.
Thank you very much, Maureen. That was great refresher, great tools and knowledge, I hope, for all of you who are listening. to avoid microbial spoilage. And that's a good transition about Brettanomyces because that's going to be the next topic from Dr. Chris Curtin. So prior to joining Oregon State University in 2016, Curtin led the Biosciences Research Group at the Australian Wine Research Institute, where he was responsible for development of novel yeast strains and strategies to mitigate wine spoilage. The Curtin lab at OSU uses omics tools to understand the role of yeast and bacteria in fermented beverages, such as beer, wine, and kombucha, where despite harboring a core set of the same species, the outcomes are quite different. A major focus throughout his career has been to understand the ecology, genetics, and physiology of Brettanomyces yeast, the preeminent spoilage risk during wine production, but a valuable contributor to beer and kombucha. So thank you, Chris, for joining us. I forgot to mention, if you have any question for Maureen, feel free to use the chat box or the Q&A box, and all the questions will be answered at the end. So Chris,
thanks for the invite to come and participate. So I can see the share button. So how does that look?
Looks good.
Looks good. Thank you.
Okay. Yeah, again, thanks for the invite. I'm really happy to come and talk about, well, despite its bad rap, it's still one of my favorite yeasts to do research on. And increasingly I'm finding that obviously it's great in certain styles of sour ale. Turns out Brettanomyces is the most important yeast one of the most important yeasts during kombucha production as well. So if you like kombucha, you might like Brettanomyces. So today, though, I'm going to talk more about, with my wine hat on, research and general insights and general strategies around the control of Brettanomyces, which has been really well introduced. And it's the reason why I think it's really good to have a focus on Brettanomyces is at the end of the day, if you've got a Brettanomyces issue, you've probably got other forms of microbial spoilage happening as well. So they all kind of dovetail together. Anyway, what I'll do today is I'll just talk a little bit about, again, why I focused a lot on Brettanomyces, some of the research that I've done or been involved with over the years that's looked at Brettanomyces strain diversity and how that informs our understanding of why it's difficult to control, and feeding out of that, but also there's a whole bunch of common sense stuff, which some of which you've already heard today. The strategies to minimize the risk of Brettanomyces and other microbial spoilages in your winery. I'll finish on that. So coming back to the general concept of wine microbiology, to start to think about spoilage, it's helpful just to think about who grows when, what's the order of the microbiology that goes on during winemaking. And depending upon what your goals are, most of the time, most winemakers are inoculating with Saccharomyces cerevisiae, and that's shown in black here. And when you inoculate with Saccharomyces cerevisiae, you successfully suppress a lot of other microbes that are present on the grapes or have entered the winemaking process on your equipment, including potential spoilage organisms, but also some non-Saccharomyces yeast, and there are even some indigenous lactic acid bacteria that could potentially have a positive impact. So again, depending on what you're trying to achieve, you may or may not be looking to suppress all of that from happening. Mostly spoilage of wine, microbial spoilage of wine generally doesn't happen at that point, although the die in some cases can be cast. If you don't have a great primary fermentation, then you're creating a much greater risk of microbial spoilage down the road. A lot of microbial spoilage happens in barrels, and it's no big surprise, barrels are harder to clean. Not shown on here is post-bottling spoilage, but I'll get to that in a second. If we're thinking about Brettanomyces, it really doesn't show up until the wine is in barrel. It might be there at very, very low numbers, but it really doesn't get going and do anything meaningful until it's got plenty of time and space to grow, and that's usually during barrel maturation. But Brettanomyces can also be a problem post-bottling if you don't filter your wine. And the way that manifests is you'll get variable amounts of Brettanomyces and variable Brett impact in different bottles, which, from a consumer perspective isn't ideal. You might taste a wine, like it, get another bottle of the same wine, not like it because of Brettanomyces. And I won't talk about acetic acid bacteria, but again, kind of showing up around the same time. It's going to be a problem in barrel, or usually only if a bung blows off a barrel, or can be a consequence of cork failure. So for the rest of the talk, I'm going to focus on Brett. So just to reiterate, what Brett is exactly. When we're talking about Brett flavor, mostly we're talking about the contribution from volatile phenols, the 4-ethylphenol, 4-ethylglycol, and 4-ethylcatechol. These are produced by a metabolism that Brettanomyces has. I think you can see my cursor. The first part here, the conversion of hydroxycinnamic acids into vinyl phenols. Plenty of yeast and bacteria, including Saccharomyces cerevisiae, can do that. The vinyl phenols aren't as big a problem. In fact, in red wines, these can actually help with stabilizing pigments. Brettanomyces is not unique in having the capability to make the ethyl phenols, but in terms of the organisms that show up regularly in wine, it's the culprit. It's the one that has that activity and is responsible. So mostly that's what we're talking about in terms of Brett flavor. It's the impact of these compounds. But they're not the only things that Brettanomyces do, and certainly if you're talking about Brettanomyces in beer, it sometimes has a very good rap for the reason that it makes certain volatile esters that have desirable fruity and floral aromas. It can also make, and this is sometimes thought about as Brett character as well, some of the volatile fatty acids that contribute to cheesiness or sweaty gym sock kind of aroma, less desirable. But overall, there's potential that if you can thread the needle of trying to get Brettanomyces to make an optimal concentration of certain esters, not too much of the volatile fatty acids, and maybe not too much of the ethyl phenols. Maybe somewhere on this flavor wheel of Brettanomyces, this was work done at UC Davis, where they fed Brettanomyces different things and recorded what kind of sensory attributes were resulting. There are some pleasant flavors on this aroma wheel, but there are also some that are not so good. So there's always been this kind of debate of, is there such a thing as good Brett versus bad Brett? It's kind of like the unicorn of the wine industry. And generally speaking, most of the time, most winemakers and most wine experts would look at Brett as a spoilage issue and not be a flavor that you'd try and integrate into your wine. But theoretically, people do try, and sometimes to good effect, sometimes not so good. So under what circumstances would you just categorically say Brett is spoilage? Sorry if you just heard my notification pop up. Because while, like anything else in wine, there's potential for things to go in balance and all be a positive contribution, I'm not particularly sold on that. In my experience, pretty much any time I've been involved in spiking volatile phenols into wine at any level, as soon as they change the flavor of the wine, which is basically as soon as you can perceive it, it doesn't really, I haven't seen a wine that process has improved. And usually it's because of this medicinal aftertaste or metallic aftertaste and medicinal flavor that comes through. It dulls the fruit and doesn't turn out very well, but don't take my word for it. Some work that I was involved in a long time ago at the AWRI, we looked at sensory responses or consumer responses to wine that we manipulated to have more or less Brettanomyces flavor. And the simple takeaway from this was, even when we only added a moderate amount of Brett flavor, consumers perceived it and it decreased the average liking score of that wine. And these, I think there were 2 people out of 101, 2 people had ever heard of Brettanomyces. So they didn't know what the problem was, they just didn't like the wine. So for me, while there might be a possibility of there being a positive contribution at the right level, I don't consider that a risk worth taking because of this kind of data. Now, could there be such a thing as a good Brettanomyces strain? And a good Brettanomyces strain would not have that ability to make volatile phenols, right? I'm going to get to that later because that's, again, one of those unicorns that researchers have been looking for. And I'll tell you about some research that has found one. So not something I would hang my hat on, but we'll see. All right. So in this next part of the talk, I just thought I'd talk somewhat about some of the research I've been involved in, which has all been about trying to figure out the factors that are important in in allowing Brettanomyces to thrive and grow. Because for Brettanomyces and its metabolism to make those volatile phenols at an appreciable level where they'll impact wine flavor, there have to be enough cells in the first place. For there to be enough cells in the first place, all of these factors have to be conducive to Brettanomyces growth. And you're all, as winemakers, unless you only use one Saccharomyces strain in your winery, you're all intuitively aware that different strains of the same yeast will respond to the environment differently. So if you use 2 strains in the winery, you might notice that one ferments really quickly and never gets stuck, and another is more finicky and you need to add more nitrogen. The same is possibly true of Brettanomyces, but I'm going to talk about the reality of that, where it does matter and where it doesn't. So I've been involved in several projects over the years to look at Brettanomyces strain diversity and try and understand how they're genetically related. And this is a collaborative project I was involved in that I'm just showing this as an example because it's the one that had the most strains. So 1,488 strains of Brettanomyces bruxellensis, which within the Brettanomyces family is the species that causes spoilage in wine. And these came from all over the world. A lot of them were from France, but there were representatives from Australia, from South America, from the US, and other parts of Europe as well. And the main takeaway here is that there are 3 major genetic groups of Brettanomyces bruxellensis that we find in wine. And we'll just call them simplistically wine A, B, and C. And the other take-home message that is relevant to how often we find different strains, or has turned out to be relevant, is that the wine A strains are able to tolerate 2 to 3 times more sulfur dioxide than the other strains. So in Australia and France, the data conclusively shows that wine A strains are by far the most common. And that may reflect the usage of sulfite by winemakers in the regions that were studied, more so than others. Hard to know, we don't really have a full handle on that yet. Couldn't tell you in the US which is most prevalent. There haven't been large enough studies to really tell us that. Can say that New Zealand, for whatever reason, has a lower prevalence of wine A and higher prevalence of the others. So it's not necessarily true that in your region that this sulfite tolerant strain is going to be most common, but it has been found in every wine region where people have studied strain diversity. So it's a very common strain that's found around the world. So in the context of this question of is there such a thing as a good Brett? Well, at least amongst these 3 groups of strains, there's just bad, bad, and much worse. All right. But I'll tell you why I brought this up in the first place. There is one study that's shown that there are such things as Brettanomyces that lack this activity, but it's fleetingly rare. The idea here being that if you had a Brettanomyces strain that doesn't make volatile phenols, then maybe you could harness it for production of some of those ethyl esters that contribute to fruity and floral aromas, or maybe at the right concentrations, their production of volatile fatty acids might not be a bad thing and just add some complexity. So this study was actually mostly to do with beer, and it was a study performed in Denmark, and they found that there was this one isolate, this strain called CRL90, which doesn't make 4-ethylphenol and had a certain gene in its genome deleted that's needed to make 4-ethylphenol. very interesting. But literally, this is the first time there's any data that suggests that there are any Brettanomyces isolates that don't make 4-ethylphenol. So 1 out of thousands of Brettanomyces isolates that have been studied to date. Worth pointing out, this is a beer strain, and it's a strain of a completely different species, Brettanomyces anomalous. So it's not one that shows up in wine. So the main takeaway here is that if you have Brettanomyces in your winery, I wouldn't hang my hat on the possibility that you've got a good Brett strain. It's just not very likely based upon all of the data that exists thus far in Brettanomyces research. So what is more important, or what can we learn? Coming back to this idea that there are different factors that might determine the ability of Brettanomyces to grow. I'm just going to show us one example, an experiment that I did a long time ago, but this was using a representative of that worst strain, the wine A strain. And what these dots represent individual wine samples so it's the same base wine. We just manipulated the composition of that wine and then looked at, after a month, how much 4-ethylphenol had been made. And this is a model that's trying to predict that result based upon the composition of the wine, the way that we manipulated it. And what we really care about is, well, why are some wines very low, or why some samples are very low in 4EP and some are very high? They've all been incubated at the same temperature for a month, all inoculated with the same Brettanomyces strain. These samples at the bottom are characterized by having a lower pH, higher ethanol content, and low residual sugar. And the flip side is true of the samples that, or the wine samples that had gone more Bretty. They were higher in pH, had a lower ethanol level, and contained more residual sugar. In this case, about 5 grams per litre, as opposed to the low being 0.5 grams per litre. What wasn't important were the factors like nitrogen and vitamins that in some past research papers had been talked about as well. You don't want to have too much residual nitrogen because that's going to be feeding the Brettanomyces, and maybe if you have different vitamin levels that aren't used by your primary yeast, then you might create more risk of Brettanomyces growing. But overwhelmingly, these were the factors. And pH itself isn't a huge factor. It's more to do with its effects upon sulfur dioxide that I'll get to in a little while. All right, but what you can take from that is, for the worst strain, the worst situation you could be in is to have a wine that is high pH and contains 5 grams per litre residual sugar. Okay, so I'm just going to move into some more practical stuff now, just talking about overall some of the insights from quite a long time of dealing with Brettanomyces spoilage issues in Australia, some of the observations and strategies that arose from all of that. Oh, actually, I have one more transition through a research point, but coming back to this question and the idea that a winery can be free of microbes has been already covered, that it's really just not feasible. You don't make wine in a sterile environment. But can you have a Brett-free winery? Well, I'd say not. And the main reason I say that is some research that was published in Italy a couple of years ago, and then some research that my lab here did over the last couple of years, where we're able to isolate Brettanomyces from the vineyard. This has kind of been a long-standing theory or idea that Brettanomyces must be in the vineyard, just like Saccharomyces is. And it turns out to be true. It's just harder to prove. And there are fewer Brettanomyces in the winery than Saccharomyces, for example. But point being, if we can find it in vineyards in Italy, and we can find it in vineyards in Oregon, based upon inference that Brettanomyces shows up in wineries all over the world, I think we can put 2 and 2 together and say, well, they're probably in the vineyard, and therefore, if you're always processing grapes, it's going to be pretty hard to eliminate Brettanomyces from your winery. So take it as a given that in your winery, there are Brettanomyces yeasts. And I've talked to plenty of winemakers who are adamant that they don't have any Brettanomyces in their winery. On the flip side of that, I can only think of one winery that I attempted to isolate Brettanomyces from, and wasn't able to. So those two things don't necessarily match up either. Okay, so just to very briefly go through some of the basics of Brettanomyces control, and then talk about some particular aspects. And some of this has already been covered, right? The very logical stuff of you need to clean and sanitize equipment, tanks, and barrels. Like if they're not clean, biofilms can form. Biofilms contain bacteria and yeast, and they create a great environment for Brettanomyces to grow. Treating your yeast and bacteria nice and getting through healthy and complete alcoholic and malolactic fermentations is really important. Alcoholic fermentations because you want low residual sugar. Brettanomyces only needs less than a gram of residual sugar to make enough 4-ethylphenol to spoil wine. It's not making it from the sugar, it's making it from the other precursors, but it needs just a tiny bit of sugar to be able to grow enough to spoil wine. So you need very low levels of residual sugar. And why do you need to care about your malolactic fermentations going through quickly? It just gets you to the point where you can add sulfur dioxide sooner and stabilize that wine. Which brings me to the next one, which is, and that's what I'm going to talk about in a little bit more detail, and that's sulfur dioxide management. It's the frontline defense against Brettanomyces. It won't work by itself, much like most of these other things that if you don't sanitize and clean everything, you're going to be throwing SO2 at the wine to try and get it stable, and it's going to be a much harder job. Barrel management is critical. I have seen Brettanomyces spoilage in wine in tank. I've even seen it in white wine in tank, and bizarrely a Riesling, which of all wines I wouldn't have thought would be very susceptible to Brettanomyces spoilage, but that speaks to the resilience of this yeast. But if you focus your attention somewhere, it's going to be on the barrels and keeping an eye on them, keeping them topped, and making sure that when you're topping your barrels, you're topping with wine that doesn't contain microbes, right? You want it to be a non-spoiled wine. A good practice is to keep your topping wine with fairly high levels of sulfur dioxide, like 250, 300 ppm, because it's going to get diluted in the barrel anyway, but it's going to reduce the risk that the topping wine you're using is a vehicle to transfer Brettanomyces. Careful and timely racking is important. Careful in the sense that you don't want to introduce too much oxygen to your wine. Brettanomyces will grow happily with very little oxygen, but it will grow much, much faster if you give it a little bump along by introducing some oxygen. Timely racking in the sense that, again, the sooner you can get to a point where the wine has low turbidity, then the easier it's going to be for you to effectively manage your sulfur dioxide. And this one should be self-explanatory in terms of not leaving barrels empty. That's more just a barrel management and the barrel's not warping and becoming difficult to use. But the shorter that window, the better. And it is very important to wash. And this is where it gets a bit tricky because I've seen examples where washing barrels has actually contributed more to a Brettanomyces spoilage issue. And it was because it was combined with something else, in this case, inadequate sulfur dioxide management. But that's why everything has to be done in combination. But generally speaking, you do want to wash. Hot water washing is, I think, better than steam because steam might, again, cause some imperfections in the barrel itself. But there are so many different ways these days to deal with a dirty barrel that go your hardest, but just don't think that rinsing with just rinsing the barrel with cold water is not going to get the job done. All right, and the important thing is to recognize when you have a problem and then deal with it promptly. And a large winery in Australia, if they had wine that started to develop a Brettanomyces aroma, it would get filtered. And they weren't necessarily in the practice of getting rid of that wine. They would, when they used it in a blend, they'd dilute it, but they might filter the same wine 3 times on its way to being utilized if they thought it had Brett. They would just keep filtering it to make sure that it, when they used it in the final blend, that it was okay to use. I'm not sure that that's what I recommend. Usually it's just better to take 0 risks. And if you do have a barrel that goes Bretti, it is possible to remediate that barrel. You don't necessarily have to discard it, but you just need to do it with an eye to being very careful that if you keep that barrel in your winery and keep using it, I'd keep it sequestered until you're confident that you've done a good job and the next batch of wine that goes in there doesn't develop Brett. In terms of how to clean and regenerate, so a good practice that has been proven to be effective is to fill the barrel with very hot water. Sorry, I don't speak Fahrenheit very well, but 70 degrees Celsius. If you can get the water, hot water, and you might need to have a steam, you might need to steam that water to get water in the barrel up to that kind of temperature, and then leave it for at least 15 minutes, or even better would be an hour, then that can be quite effective at the heat penetrating into the staves enough to substantially reduce the Brettanomyces population. It's also been shown that scraping and firing barrels or retoasting the barrels, unsurprisingly, again with the heat, you're able to effectively remove a lot of viable cells from a barrel. Again, there are a lot of other options out there as well. All right, so briefly, a little bit more on sulfur dioxide management, because it's really important, and particularly in light of the fact that some strains of Brettanomyces can tolerate more sulfur dioxide. If you have one of those in your winery, then you really want to be on top of your game with sulfur dioxide management, or be willing to use alternatives like chitosan. So in terms of overall sulfite management, and I'm not going to talk about pH specifically, but go back to your basic charts that show molecular sulfur dioxide and the relationship between that and wine pH, and we'll get to that in a second. But starting with the fruit, unless you're trying to carry out spontaneous fermentation, then definitely adding a decent amount of potassium metabisulfite either into the picking bins or at crusher is a good idea, and more if the fruit is in any way damaged or diseased. And that's going to help with managing not just Brettanomyces, but a whole lot of other potential microbial issues. So if you want to do a spontaneous fermentation, allow the ambient yeast to take over, you may want to dial it back a bit, but just be aware that that is taking a risk. And the important one is that in a red wine after MLF, that's when you want to make it your largest sulfur dioxide addition, and you want to try and achieve, again, referring to your wine's pH, a molecular sulfur dioxide level of at least 0.6, but preferably 0.8. And if your wine pH is 3.5, then that 0.6 ppm of molecular sulfur dioxide translates to 30 ppm of free. And that's why if your wine is close to 4 in terms of pH, you really aren't going to be able to rely upon sulfur dioxide for microbial control. It'll still work as an antioxidant, but it's not going to help you for antimicrobial efforts. Some things to remember: that when you're adding sulfur dioxide or adding potassium metabisulfite, then only 35 to 40% of the sulfur dioxide is going to be yielded in the free form. The rest will get bound up. And while we need to care about total SO2 for lactic acid bacteria like Enococcus, really it's just the free and molecular part that works against Brettanomyces. Clarifying wine is important from the point of view that if you have a very turbid wine, when you're adding sulfite, you're going to get less in the free form, more of it's going to get bound up. But the takeaway here is that it's all about timing and magnitude, and it is about pH management as well. You can't think about SO2 without thinking about pH when it comes to microbial control. So the closer you are to 3.5, the more chance you have of being effective with your additions. Again, why is all this important? So I talked about the fact that Brettanomyces, or that wine A strain is already more tolerant than other strains. Some research that we did back in Australia, where we took different strains of Brettanomyces, in this case, representatives of the wine A, B, and C. Ignore the annotation here of A, B, and C. That's referring to the replicates, but that's not important. The important point here is we took these strains and we put them in the presence of sulfur dioxide and then gradually increased it. So each time we grew it, we added a little bit more. And over time, we were able to grow them at much higher concentrations than the strain could tolerate initially. If we then took isolates from that endpoint of the experiment and we did an experiment where we asked, well, what is the maximum amount of sulfur dioxide they can tolerate? So you can see this blue bars here correspond to wine A after this experiment. This bar here corresponds to wine A before that experiment. You can see that it already is pretty close to being able to tolerate 0.6 milligrams per liter or ppm of molecular sulfur dioxide. After the experiment, the isolates are tolerating much more, even above 0.8. Why is all that important? Well, if you're adding small amounts of sulfur dioxide in your winery at a time, then you're kind of doing this experiment in the field. You are training whatever doesn't die to grow in the presence of more. And that's where you can definitely run into problems with never being able to keep up with the spoilage issue. So that's why the large additions. So if we added a large addition at the start of this experiment, the yeast can't grow. If we add a large addition at the end of this experiment, it's not going to be as effective. We might still see some growth poking through. So that's kind of like a cautionary tale from current research to when you're managing your sulfur dioxide, think about it as fewer large additions rather than a lot of small additions. So hopefully that's been an interesting story of weaving some research into some of the key things we talk about for Brettanomyces control. But just to circle back, some of the key things to take away that Brettanomyces it's not the only spoilage issue, but it's impactful. And if you have Brett issues, you probably have lactic acid bacteria spoilage going on as well. So they kind of all go together. Focusing on Brett is not a bad way to start dealing with microbial spoilage. I just encourage you to see that there's very little chance that if you have Brett in your winery, it's going to be a good Brett. There's very few examples of any wines being improved by the addition of Brett, so I err on the side of caution. And some of this research is highlighting the potential for emergence of sulfite tolerant strains, so making sure that you're doing those large additions rather than lots of small additions is a good idea. Other than that, mostly it's focusing on best practice winemaking, and that will take care of a lot of spoilage issues. If your winery is clean, your equipment's clean, your barrels are well managed, you don't have bungs popping off here and there, you don't have wine sitting on ullage, a lot of spoilage issues are going to go away if you're managing all of those things. And the last thing I'd say is that if you do think you have spoilage, then getting onto it quickly and segregating that wine, trying to take it out of the environment where all your other wine is being stored, or at least you're marking those barrels as being potentially suspect, then you can minimize the impact. The worst thing you can do is wait and see what happens and end up spreading a Brettanomyces infection around. So with that, just finish with a very 2021 lab photo of my students from earlier this year. Thankfully, we're getting closer to the end of that. And I'll turn over to questions. Thank you.
Thank you, Chris, for this great presentation. If you didn't type any question, I don't think anybody asked question for the moment. So you still have time for questions in the chat box or the Q&A box. Otherwise, if one of you has any question, feel free to ask. I have some questions, but...
As do I.
Okay, you want to start, Drew?
Sure. First of all, can winemakers get their spoilage bacteria tested at the lab at ISU?
Not currently. It's something that we have talked about whether there's enough demand for it to justify the material cost, basically. And it, you know, every so often it comes up. And so it's definitely, you know, we have planning meetings occasionally and we go, okay, is it time? Have we reached the threshold to make it worthwhile? So no, I'm currently still directing people to one of the larger commercial labs who can do, you know, the 2-day turnaround on your PCR test to identify, you know, whatever spoilage you have going on.
But you can check acetic acid, volatile acidity. That's one thing you can check.
Yeah, all the chemical, you know, the chemical indicators, the chemical
Right.
red flags.
That's what I meant.
For sure, yes. Yeah.
Okay. I was going to make regarding filtering acetic acid out. Are there any? Do you know of anybody in the Midwest who has a mobile VA filter at all?
Not mobile. There's one person, one that I know of. Jenny actually would know better than I would, so maybe she's watching and would text me or something. No, I'm kidding. There's one winery that I know of that can do VA removal. It is not mobile, but I think they've done some custom stuff where people will, you know, ship it in a tote over to them and they'll treat it. And it's possible that there are other. I'm just thinking of one in particular. And if anyone is interested in information on that, please feel free to reach out and I can work on putting you in contact with them. I don't necessarily want to call them out publicly, but
Sure. You made a really good point about rotating sanitizers, and this is really important because most winemakers, it's not widely known or widely trained, at least any of the commercial wineries where I worked, to rotate sanitizers. Do you have any idea how often, especially like during harvest, would you go 1 week and then 1 week sort of thing, or every day, or?
I've heard varying practices here, and maybe this is something where Chris has some input in terms of building resistance. The practices that I've seen put in place are actually more like, you know, half the year is one, you know, so 6 months on one and rotate to the other for the other half of the year. But I know of people who are doing, you know, every week they switch or every 2 weeks they switch. That's just not my personal experience with it. So yeah, if others have input, I would love to hear it.
I have Nina who raised her hand, so I'm gonna unmute you, Nina, if I can. Wait a few minutes, because maybe that's for a comment or a question. Nina, you're unmute.
Yeah, I got a quick question. So this is Nina and Ron Harvey from Maple Ridge Vineyard. I haven't heard anybody talk about what we call film yeast that kind of builds on top of tanks. Is that some form of Brett or something that in the winery, or what would that be?
Can be, but it can be multiple different. It could be made up of multiple different organisms. There are specifically film-forming yeast. Often Pichia is one of them, and yeah, their impacts can be from minor to significant. There are some Pichia that actually make volatile phenols, but they generally don't get too much opportunity to thrive. Usually the film-forming yeast like oxygen, so you're getting a film that's an indicator maybe of too much oxygen.
Okay. Great, thank you.
I don't like sulfur dioxide, I'll tell you that much.
No, well, I'd like to limit it, but you know, sometimes that's the only option. But I'm kind of, we were talking about using some inert gas of some sort, but you were mentioning that that some of the Brett may be a problem for that. So I don't know if I want to encourage that or not, but especially
Brettanomyces will grow faster aerobically, not anaerobically. So if you're doing anything to try and control, you know, oxygen liking film forming yeast, it's not going to hurt in terms of Brettanomyces. Like, you're not going to encourage their growth.
So some inert gas would probably be the best way of stopping the film yeast and things like that on the surface.
Yeah, assuming it's yeast. So it is important. It's not a bad one to check, possibly by the PCR analysis that was talked about, because it'll tell you if it's a bacterial or yeast film. It's probably
Okay,
yeast.
great.
I'd like to add a couple comments. If you are noticing film yeasts, it's as Chris mentioned, there's no problem at all with doing a preliminary filtration of the wine, even if the wine is still undergoing maturation. You could go right ahead and do a filtration. Of course, a big shock dose of SO2 is always a good idea. Regarding inert gases, there's really two choices, nitrogen and argon. Nitrogen is less expensive, but doesn't last quite as long. Argon is more expensive, but it seems to last. It's not only heavier than nitrogen, it just seems to last longer. So the commercial wineries where I've worked, if we're using nitrogen, we would sparge the headspace on a partially filled tank with nitrogen twice a week. With argon, we just do it once a week. If you notice a film yeast and you can't get to it right away, you can't filter it right away. I worked at a Gallo winery where we would just take a spray bottle and fill it with some very strong, like 500 part per million solution of potassium metabisulfite and just spray it. Open the tank door and get in there and spray that surface, and then hit it with either some the headspace with some nitrogen or argon, and then put the filtering of that wine on the schedule as soon as possible.
And to echo what you're just saying about sparge or adding a lot of SO2 just to maintain or to reduce the risk of microbial spoilage, Chris, what do you think about alternatives of SO2? Because you just mentioned that some bread strains start to be tolerant to SO2. So would you have any experience about an alternative that would work in addition to SO2, or just to remove completely the use of SO2?
As an adjunct to it, so chitosan products like Lallemand is called no bread inside. I'm not sure what but I'm sure they have equally exciting names, the competitor products. That I know Lafort have one. I'm sure there are others out there now. And I've seen both research and real world examples of those kind of products working effectively. And there does seem to be, again, some differences between strains as far as how effective it is, but I wouldn't, not quite to the same extent as sulfur dioxide. So it does seem to work quite well. It works obviously differently to sulfur dioxide, so that's something you can use at the same time if you're particularly worried. It's an additional cost. So then working out where and when to use it. But it can, I've seen a very active Brettanomyces event in a barrel stopping its tracks with the addition of chitosan and no bread inside. The increasing 4EP just stopped, and it held, like it didn't, trying to think how long it went out for. It was at least 3 months, possibly 6 months, that it stayed at the level that it was without doing anything other than adding the no bread inside.
Okay, thank you. And so if I can, we still don't have any question. Feel free to use the chat box. I still have one question. So when you mentioned that there is a higher concentration of 4 ethyl phenols when you have high pH, low ethanol, high residual sugar, what would you be thinking for grapes that we have in the Midwest, where we have low pH, medium ethanol, I would say, and high residual sugar? What do you think would happen?
In that context, the low pH and Brettanomyces will grow. I've grown some strains at pH 2, so the pH itself isn't enough. pH in combination with low pH in combination with ethanol becomes a little bit more impactful. But the reality is, if you have something that's sitting in barrel in particular for a lengthy period, then there's a lot of opportunity for Brettanomyces. Even if it's not growing optimally, it'll just slowly, the population will increase. And the residual sugar is definitely a problem, but unless you can get the wine bone dry, less than 0.5 grams per litre, there's risk of Brettanomyces growing. So if you accept that there is, if you're above that threshold, then you need to assume that the conditions are going to be conducive. Because unless you're getting up to, again, I isolated Brettanomyces from a wine that was 17% ABV. I think that's the highest I've seen. So, it's pretty hard to ever look at a wine and say, you've got a low enough pH, a high enough ethanol, of just those two things to be enough to be inhibitory. If you've only got, 0.1 grams per litre of residual sugar, the potential for a spoilage event goes down substantially, but it's really hard for that to be the case.
Yeah. And did you also look at the level of hydroxycinamic acid you had in the grapes? Because is it really related? Like the more you have, the more for ethyl phenol you would have. So do you have an idea of differences between different grape varieties? Because all varieties are different in terms of that.
That's a very good point, one that I didn't bring up, but thanks for bringing it up, because it's true. It's just one of those things that at this point, I wouldn't encourage anyone to get too worried about, because we don't know enough. But I do know categorically that if you take the exact same wine and you double the amount of the precursor, you will get double. Nothing else changes. The Brettanomyces grows the same. You get twice as much 4-ethylphenol. So there is an absolute direct correlation between those two things. It's just not something that we know enough about different varieties to be able to say, well, this variety is really low risk. We do know, for example, that Pinot Noir has a very different phenolic profile. And if Brett grows in Pinot Noir, you get a very different balance of the 4-ethylphenol, 4-ethyl glycol, because of the differences in those precursors. So there are some varieties that we can point out and say, we know that phenolic profile is very much related to how the Brett ends up looking. I just don't know if there are any varieties where, well, hydroxycinamic acids are going to be in pretty much everything. They are going to be at different levels. It does contribute to the risk factor. We just couldn't tell you which ones are good, low risk.
Yeah. Okay. I think that's good. That's good for me, because more research is important.
Yeah, it's definitely, there might be some varieties that are much lower risk than others based upon their phenolic profile.
Yeah. Thank you.
I have
Anybody?
another couple of comments I'd like to make. I'll keep them brief. Chris, I really, your presentation was really thorough and complete, and I thought I knew all about Brett. My question before you finished was, can non-barrel wines or wines solely made in tank get Brett? You answered that. The answer is yes. Maybe not as widespread, but it can certainly happen.
Definitely less frequently, yeah.
Yep, and then you made another great point about an immediate post-fermentation, or if you're doing malolactic, a post-secondary fermentation shock dose. If I can borrow that from people adding shock chlorine to their swimming pools. I worked for 3 years for a Gallo cellar in California, and boy, every wine, when the white wines were finished with primary, they got if they were 3.5 or below pH, got a 60 part per million shock dose of KMBS. Likewise for the reds, once they had finished primary and secondary, if the pH was much above 3.5, we even increased it to a 70 part per million dose. I loved your suggestion about keeping your topping wines very high. You said 200 to 300 part per million. Of course, they will get diluted, and certainly that is making it very difficult for virtually any spoilage bacteria.
Just don't mix up that barrel and forget about where that goes and end up putting it into something else, because that would not be good.
That would not be good. or even just people topping wine in kegs or in carboys and such.
Yeah.
Give them a good strong dose before you put them away. The only other thing I was going to add is I worked at a winery where we had a very active Brettanomyces management situation. And when a barrel was identified to have Brett, we would paint a big purple bee on the head of the barrel, like a big one. And then we also had special, I'm sure you can get them, they're out there, but it was a colored, not just a natural silicon bung. This was a purple bung. And so all of the B barrels had a purple bung on them. And that would, this was a large winery that had a dedicated lab person, and that would remind that person to be careful when they were sampling the barrels, to remind them visually, to be careful about cross-contamination. So if you do have Brett barrels and you want to manage them, make sure you mark them. You don't want to cross-contaminate with them. And
really good.
as for the most perfect way to clean a barrel, I like steam, but you're absolutely right. How it's applied and the pressure it's applied, it can possibly damage. Yeah, can sort of open up the grain of the wood and make nice little homes for even more Brettanomyces. Being able to fill a full barrel with 70 centigrade water for an hour, that's a lot of heat and a lot of water. Not everybody can do that.
It's not super sustainable. I mean, I will say that you can, if you had 3 barrels, the temperature of the water is not going to change much. You can pump it from one to the next. Probably not the most fun thing for your pump to be doing either. And obviously there's health and safety issues associated with very hot water. So
Yep. No,
trade-offs, but
indeed, isn't winemaking always a system of trade-offs, or often, not always, but often. One other question for either of you, Maureen, maybe you've got an answer. I'll just remind the audience that the Midwest Grape and Wine Industry Institute at Iowa State University does a wonderful tasting proficiency course. And one of the parts of the course is that they give you samples of wine that have slightly above threshold amounts of all of the fault indicators. So whether it's VA, acetic acid, or excess sulfur, or oxidation, or Brett, you can sort of try to train or guess what the fault might be. So Maureen, how can somebody train their nose if they're not familiar with Brett? How would you suggest a winemaker train their noses to be able to spot Brett more easily?
Oh, that's okay. So that's the way that we do it for our sensory training. It's a smell only. It's not one that people taste. So we dose it with 4-EP, and 4-EG. And you know, that's a hard thing to get your hands on as just a generic consumer. And again, it's not something that you want to be tasting. So, I mean, this is my personal approach is, you know, look at the typical descriptors for Brett, right? So you can have smoky characteristics, meaty characteristics, leather, medicinal, banded. I mean, you kind of have to break it down, I think, into its component descriptors, and then you can actually seek those things out and smell them. And this is what I do, honestly, with any sensory training where, you know, if it's fruits and vegetables, I will go around and sniff the produce section. So my personal recommendation would be, you know, you're not going to get your hands on 4-EG and 4-EP, but you can get your hands on some of the other things that we use to describe Brett characteristics. Short of that, I would recommend going to your local wine shop and telling them that you actually like a more, you know, the rustic Brett characteristics in your wine, and they can probably find you one that has Brett characteristics, and then you can actually smell and taste it in the context of the wine, which you cannot do by when you dose it. You can't be tasting the wine when you actually are using its non-food grade chemical compounds that, you know, us mad lab scientists work with. Yeah, you can't be tasting it. So just go to your local wine shop, tell them you like a little bit of a Brett character, and let them help you out.
Or then some kits exist for just wine aroma, but also wine folds. So if you can purchase the kit with wine folds, I think that's a good training as well.
The only thing I'd add to that as well is, if you know your wine, you know what it looks like, and be on the lookout for any barrels that you're tasting through and notice that there's a lack of fruit, because that's the first thing that you'll pick up is more, you won't necessarily perceive. It's a very quick transition between the two, but you might not necessarily immediately perceive it as Brett, but you'll notice that that wine is less fruity, and that's a good indicator. And the other indicator I was going to mention that doesn't rely upon sensory, that some large wineries in Australia use very successfully, was if you're already measuring free and total sulfur dioxide routinely, then the ratio of those two can be an indicator, because if you have a lot of microbial activity, you might have 60 total, and very little of that is free sulfur dioxide. It's mostly bound, and that's again, it comes down to baselining your own wine and looking for the sample that really sticks out. And if it's sticking out because it has a low amount of free relative to the total SO2, that's a good one to go look at from a microbial point of view.
And I think that's something I don't know if you would be like, if somebody would be able to use consumers, but in one of the graphs you presented, the relationship between the consumer liking score with the Brett flavor, probably you can ask your consumers if they like or if they don't like your wine. I'm just kidding. But
that's true. But
I like,
consumers, I think the surprising thing about that study was the correlation between consumers and what I didn't say was that other axis was a trained sensory panel rating the amount of Brett flavor. So a trained sensory panel versus consumers who don't know as much and certainly don't know about Brettanomyces, the correlation between the two is pretty strong.
Yeah, so that's just saying that you should make sure you don't have microbial spoilage or Brett.
It's always a good place to start.
Yep.
Difficult to achieve when, as we know, winemaking is not sterile, and that wine is a great environment for a lot of yeast and bacteria to grow. Yep.
All right. Thank you very much. Does anyone has any question? Everybody is ready for harvest? I don't know.
I'm not quite ready for harvest, but I'm getting there.
Okay.
And I could just mention a few things for practical cellar for getting ready for harvest. If you have any new people working in the cellar, take good care of them and make sure they know not to walk backwards, that hoses and sharp edges exist, and to never be in a hurry in the cellar. It's a great time of year to take your pump head apart and clean out your pump head. You might want to have a spare impeller on hand. And if you use a forklift, now would be a good time before harvest starts, if it hasn't started for you yet, to get an annual maintenance on your forklift. There you go. Thanks, Aude.
Thank you. So I would thank you both, or all of you, for being with us today to present about microbial spoilage. I hope it was helpful for all of you attendees. If you have any question, feel free to reach out to Drew, myself, or directly to Chris and Maureen for any question related to that, to be ready for harvest. Keep in mind that you have a QR code for a survey. If you don't have access to the QR code, I put the link to the survey on the chat box. So please take 2 to 5 minutes to complete the survey. And also, here's the list of the future research and winemaking webinar that will happen starting in October 5th. We don't do anything this summer, just to provide enough time to everybody to focus on wine. And so we're going to do sorbic acid. That's going to be more round table, more than an event as we did today. And then the next one will be November 2nd on bentonite treatments, and then December 7 on oenological tannin. And also something that probably you know, we have town hall meetings gathering viticulture and oenology that is happening between a lot of different states, probably all the states of the Midwest and Eastern US. So feel free to register for this meeting. It's going to happen next week. You can submit your question in advance. And the topic would be for viticulture, Verizon to pre-harvest, and oenology would be focusing on winemaking additions. So feel free to register to that town hall meeting. Once again, thank you very much to all of you. Thank you for being with us today. And I would say have a good day. Have a good summer, end of summer.
Thank you.
Bye, everyone. Thank you very much.